we've seen some interesting talks about the actual science that people are doing with our systems. And I wanted to take a step back and take a look at the technology that underlies this. I realize that a lot of people here maybe are not as familiar with multiphoton imaging or the different methods that we use for multiphoton imaging, for optogenetics, for example, or for large field of view imaging, for deep imaging, and for, of course, some of the things that I'll talk about at the end, ultra-fast imaging. So just really briefly, Bruker fluorescence microscopy consists of several smaller subdivisions of different companies that Bruker has acquired over the years. I specifically work with multiphoton imaging, as Sarah mentioned, which comes from the original Prairie microscopes, purchased quite a while ago at this point, super resolution microscopes, light sheet microscopes, miniscopes and multi-region scopes, and you'll hear more about new miniscope technology later in this session, of course. Data storage and analysis solutions, and of course, spatial biology. But again, I'm going to be focusing right now on multiphoton imaging, as that's where I work and where I have a history in science. Those of you who are totally unfamiliar with the technology that we use, it's a scanning microscope. So it's very similar in concept to confocal microscopy, if you're familiar with that. In one photon confocal microscopy, illumination occurs across like a cone, essentially, of light with a focal plane and a pinhole that is also used for descanning for optical sectioning. In two-photon microscopy, we use near-infrared light, and this spot is scanned just as it would be for confocal microscopy, but we're using light at a longer wavelength, and fluorescence only occurs at the where there's enough photon density for emission. And that actually only happens at a tiny spot, meaning that optical sectioning can be done simply from illumination. So we don't have to descan our image as you do with confocal microscopy. And that means that all light that is gathered or generated during scanning can be collected even as it scatters through tissue. So the laser itself can penetrate tissue much more readily as it's longer wavelength, and emitted photons are more likely to reach your detector as we can collect scattered light from it. And so for that reason, this is widely used for in vivo imaging and also for two-photon optogenetic stimulation, as there's no off-axis effects, and stimulation is defined just by a small, often diffraction-limited spot. And when we're considering what we want to do for efficient microscopy and functional imaging, is we want high resolution. So we want to generate a spot that is as small as possible. We want to have as large a field of view as possible for imaging as many neurons as possible. This is one of the major advantages of in vivo imaging over, for example, electrophysiology, in that you can see large areas, hundreds, potentially even thousands of cells at a time. We want to image with a frame rate or a volume rate that's sufficient to capture neural activity. I'll talk a little bit more about that later too. And we also need to penetrate deep into thick scattering tissue in vivo. And ultimately, we combine this with our optogenetics tools and synchronize this with behavior, potentially with patch recording, or with stimulus, such as virtual reality systems. And just a quick example of what this sort of looks like. At the top here is one of our more recent microscopes, the 2P+, with a little track here for a mouse, so the mouse can run around on a track, synchronized with imaging. This bottom was actually from my postdoctoral lab, where a fly can be mounted on in a virtual reality arena and can walk around on a ball. And all that can be synchronized with your imaging data. The Prairie system started quite a while ago, and we were an early adopter of the technology behind two-photon laser microscopy, licensing the patent early on and generating the first sort of Prairie Ultima in vivo. Some of you might even have seen these out in the field. They're still out there. imaging and photostimulating. And what was really innovative about this, and still is today, is the multi-path imaging setup and photostimulation. So independent sets of galvos control an imaging laser, and a separate set of them control a photostimulation path, meaning that you have independent control of where you can target while imaging. And of course, most modern scopes have a resonant galvo path for higher speed imaging. And this started out really with glutamate uncaging historically, but has now expanded to a variety of methods of optogenetics, so activation, inhibition, potentially even photoablation. All sorts of different methods can be used with this technology. Now, this has, of course, developed into and evolved into our current workstation, the Ultima 2P Plus. So this microscope is developed to have a large field of view and greater collection efficiency, so wider angle of collection, meaning sensitivities is great at deeper tissue levels. It's compatible with longer wavelengths of laser, so it can be done with 3P imaging potentially if you so desire. And it has a good, quality field of view, so you have your good image across a wide field with uniformity. And it also has the ability to work in 3 dimensions with our ETL and our spatial light modulator, which I'll talk about in a second. What it does is, like I said, these large field of view and large optics mean that we have a 2 millimeter diagonal field of view with a commonly used 16x objective, making this ideal for large-scale calcium imaging. And of course, wide collection apertures are optimized for collection in deep tissue. Now, what we often talk about, if you've seen some of the talks earlier, where people describe using either the Neurolight 3D or spatial light modulator, is our holographic stimulation platform. So this allows you to stimulate in 3D across hundreds of microns of space. It produces really high resolution spots. So you can get single cell activation and even subcellular activation, depending on your setup. It has fast switching time, so you can generate 600 holograms per second. So you can switch different patterns to stimulate different sets of neurons. And of course, it allows you to image simultaneously when you're using decoupled focusing modules like our ETL, which I'll also mention. And what this looks like in real time, of course, is that you can select subsets of neurons at your own desire, stimulate them in either 2D or in 3D. Here showing a couple stimulation planes and imaging multiple planes at the same time while stimulating across them. So it really truly allows you to do 3-dimensional imaging and stimulation. And it integrates really well with our software, where essentially you can define cells here just by clicking on them, defining the area of activation you want, defining the pattern and power, and then generating a group with that to stimulate subsets of neurons. So it's really, truly kind of a user-friendly method for doing really complicated, what used to be really difficult experiments. We also combine this with our scripting API. So everything that you can do in our software can be scripted, and everything that can be scripted could be run using Prairie Link. So if you're so inclined to generate closed-loop optical experiments, you can read images as they're being generated. You can do online analysis, and then you can send commands to the scope. So, for example, moving the stage or generating stimulation patterns, changing powers, changing wavelengths. All these things are possible for advanced closed-loop experimentation. And as I mentioned before, we typically combine this with our electrotunable lens. These are also sometimes known as liquid lenses. These are variable focusing lenses that are in the light path that allow you to decouple imaging depth from the position of your actual objective lens. It also can make really fast jumps, so you can move quickly across large distances of tissue without too much delay in your imaging. And of course, we also have specifically patented and created optics for this to generate a consistent point spread function across the entire Z range of this. So it's, some ETLs out there, I won't say anything too much about them, but often have issues with their point spread function degrading at the edges of their range, but we correct for this with our system. And we also correct for temperature compensation and other various issues that people do encounter with other ETLs to make a consistent quality imaging across its range. And functionally, what this looks like is this, we're allowed to now stimulate in 3 dimensions without actually moving the objective. If you compare this to what we traditionally do with a fast imaging device or a Z device like a piezo, the piezo moves the objective up and down, changing the focal plane. This also alters your stimulation pattern as well. Whereas ETLs allow remote focusing, meaning that you can stimulate in 3 dimensions as well as image without disrupting the localization of either. And it's compatible, of course, with a wide variety of imaging methods, including through grid lenses. So I will just quickly highlight here, if you're interested, we have a webinar from a postdoc in the Bruker lab at the University of Washington describing SLM stimulation into the amygdala through a grid lens. So it's compatible with a wide variety of imaging methods, not just for cortical work. So if you need to do deep brain work or anything like that, you can still use this technology. It also is compatible with our relatively new focusing device called the Neuroscience. This is our ultra-fast focusing and depth control device. So this uses a DMD mirror, so a digital micromirrored array, basically allowing us to switch focal planes by just turning these mirrors that are ultra-fast, meaning that we can switch focal planes in under 20 microseconds, which for the purposes of our imaging here is essentially instantaneous. And this means that you can generate Z stacks with no delay, and you can make large jumps with no delay. It also allows you to control the depth of field as well. So you can elongate your depth of field from a smaller, tighter space, like 10 microns, to 80 microns, or depending on how you set it up, even potentially hundreds of microns. So you can collect whole volumes potentially at just single frame rates using this. And yeah, again, this is also, like I said, remote focusing device, so it doesn't change your position of your objective, making it compatible with our optogenetic platforms too. But yeah, so I want to talk a little bit about some of our new developments as well, and I'm going to pull back a little bit to talk about just when we think about measuring neural activity. Traditionally, for a long time, neural activity was predominantly acquired by electrophysiological methods. So I also did some of this as well before I got into calcium imaging. It's a tried and true method. You can measure spikes, you can measure subthreshold events, and you can measure this with high spatial and also high temporal resolution. It is somewhat limited though, depending on the method. You can only you can only collect from a few cells at a time typically, or potentially maybe more if you're thinking about a neuropixels type device. But you don't have the ability to genetically restrict or control exactly what cells you're looking for, like you can with genetically encoded indicators like GCAMP. So there has been some desire in the field to kind of bridge these two methods using voltage indicators, as calcium imaging is relatively low temporal resolution measurement of neural activity. There is, the goal here is to actually measure with high fidelity and high speed the voltage of an actual cell. So this is where voltage indicators have come in. Now, there's a lot of development has happened in this field too. I also remember the bad days of very poor SNR or having to use dyes instead of genetically encoded indicators. Of course, the advantage for genetically encoded indicators, as I mentioned, is for cell specificity, and various other reasons why you would want to use genetic control. But yeah, voltage indicators have now have reached the point where their FNR is getting high enough for most imaging systems to work, but they have been historically dim. They do tend to be harder to image because there's membrane localization. You can't completely overpack membranes with these or they disrupt cell function. And of course, it requires imaging technology that can image at the speed of action potentials, meaning that you need to be able to resolve something that might be only a few milliseconds long in time, depending on the indicator that you're using. So what we've done to get around this is develop this module called the optivolt module that allows us to do kilohertz imaging rates. So this will make it suitable for voltage imaging, neurotransmitter probes, even some vasculature work that requires extreme high-speed imaging. It's modular, so it's an additional part to an existing microscope. It is, of course, also compatible with our 3D optogenetics. So holography is compatible with it, and all of our optogenetics methods are compatible with it. And we've also developed a brand new custom FPGA-based digitizers for this to help with the collecting of the low signal that results from small dwell times and fast imaging rates. And we developed this in collaboration with a group at Boston University. And what this does is it relies on the already existing resonance scanner that's in most of our microscopes already. And we use a custom set of micro lenses in an array. And each one of these lenses changes the scan angle of the resonance scanner. So we haven't sped up the resonance scanner. We're just using an optical trick to make it scan faster, essentially. So now every line of resonance scanner, if we go across our 16 lens array here, produces now 16 lines of scanning. This also allows us to keep our point scanning technology, right? So we can still image deeply into tissue. We can still collect scarce photons, and we can collect in wide angle efficiencies. What this looks like on your sample, of course, is that as you would have, as I said, one scan of this resonance scanner would produce 16 lines per sweep. It is a somewhat limited field of view, and I'll show you a little bit more about that. But it allows us to capture now over 1,000 frames per second of an area of interest. And it integrates really well into our software. So if you just are imaging here, something in resonant mode, you collect your wide field of view if you want, define where you want to go, adjust your P&T settings, all the normal things that you would typically do. And then you just engage the optivolt module, move around your field of view that you want, and to enter that, and then you can start imaging there. You can actually see the field of view here move around. The field of view is smaller if you want to do faster imaging and potentially larger. So about 200 microns across with a 10x objective at kilohertz. Good. And a little over 400 microns across with a 10x good objective at the 500 hertz mode. And you can toggle easily between these two modes depending on what you need. Good. Of course, like I said, this can be freely moved good around, and eventually our goal is to have this be tileable so that you can automatically image a larger field in succession. And of course, if you're doing this in good combination with optogenetic stimulation can occur anywhere in your overall field of view because the galvos that control stimulation are uncoupled from this good path. So we have the full field of view available for optogenetics. And you would, just like you would for any kind of scan here, set up your T. So it does really, the optivolt allows for high-speed imaging. It's compatible with current resonant traditional galvo modes. There's just software to toggle it in and out. It retains the high-resolution point scanning architecture so that you can still image deep. And it also works, like I said, with calcium imaging, vasculature imaging, neurotransmitter imaging. And it also produces, a single square image. So it's compatible with all of your like workflows that you already have maybe developed for just your slower imaging calcium imaging. And ideally, this is going to be functional, with our systems in the future. Right now, there's only a few of these out in the field right now, but it does work well with our SLM, and it does also capture spikes in real time. Here's an example from one of our demo systems installed at Stanford, recording of an ASAP cell. But the goal is now actually to get this working with true functional mapping across large brain areas of the brain. So if you're interested in this, let us know, and we'd love to talk to you about working with you. And just to basically summarize all the things that I talked about right here, we work in multi-photon imaging to produce high resolution, large field of view, and images at high frame rates with good SNR. This is ideal for making multi-photon imaging accessible to everyone, really. It's not as hard as maybe some people think it is. You can image deep into tissue with it. And of course, you can easily synchronize with your behavioral setups, your stimulus setups, and even electrophysiological setups. It's compatible with ultra-fast focusing devices like our Neuroscience, our holographic stimulation, like the Neurolight, and of course, our new imaging module for high-speed voltage imaging and high-speed various other imaging methods. So with that, I'd love to, I'm definitely going to finish on the early side for this, but I'd love to take some questions. So yeah, let's go ahead and do that. Thank you.
All right. Thank you so much, Kevin. Wow. The ability to define cells by clicking on them to curate that subset of neurons for stimulation is really a dream. And then taking it then through a grid lens. I understood that correctly, right?
Yeah, that's correct. So there are definitely some challenges using it with a grid lens, but there are a couple labs now that do this. This is from the University of Washington, but 𝗡𝗘𝗨𝗥𝗢𝗦𝗖𝗢𝗑 lab at Salk is also using stimulation through a grid lens as well. And I don't really know how far along they are in publication, but I have seen some of their data, and it does look great. So it does work.
Yeah, that just opens up so many avenues for brain manipulation. It's really cool and powerful to combine.
Yeah, I was going to say too that there are some people out there, Sarah, that I'm sure you know as well, who are even taking this, taking a mouse with a grid lens embedded and doing mini scope imaging, and then also potentially imaging it as well under a 2D scope. So it should be totally compatible, I think, with the two.
Yeah, that's a really powerful workflow. And you're right, there's a few pioneers really pushing the limit in that area.
Of course, with the new mini scope.
And we already heard a request yesterday from one of our speakers about being able to do 3D optogenetic stimulation in a free behaving animal. Definitely a future direction, but an exciting request. So we have a question here, and this is really related to, I think, pushing the technology even further. So they ask, is it possible to make a field portable, miniaturized, super resolution, two-photon microscope that can image tissues like brain without losing information to the light and matter interaction? And then they clarify further that they want to go beyond, almost take it beyond Abbe's diffraction limit. So what do you think about that, Kevin? Is it possible to make a field portable, miniaturized, super resolution two-photon microscope for imaging brain?
Wow, I've never been asked that before. I am not familiar with how two-photon light would work with super resolution. Yeah, I honestly maybe this should be something I should ask some of the super res team, but as far as I know, we don't have super res microscopy is not really great at tissue penetration per se. It's kind of limited in how deep you can actually image with those. So if you want to go full super res, I'm not really sure what the solution is there. That's definitely beyond my area of expertise. As far as making these systems smaller and more portable, though, that is definitely happening. Mostly because, so I mean, if you see these things installed, right, they're actually they're quite massive. And there's a reason that we have this large footprint, huge microscope, and that's simply because it's a lot easier to optimize optics and a lot easier to customize the microscope when it's large, because we can change glass elements in it, change other elements into it to really customize it. But what's happened now is fiber coupling to two-photon lasers has reached the point where it's actually good, and dispersion is not as big as a problem as it used to be. You are limited by certain things when you start fiber coupling. For example, if you want to use really wide aperture lenses, there's issues of beam size that go into this. But when it comes to like miniaturizing this, that has actually really been a boon, because you can the ability to fiber couple with a 2P laser means that you can actually have something moving. There are some people out there who do tracking 2P lasers, but this is something that's probably very difficult to maintain in our custom-made kind of rigs, where you basically have an objective lens kind of on a modified CNC mill top that will actually track along with an animal, but it's notoriously difficult to do. So I don't really know if there's a lot happening in that field. But yeah, as far as miniaturization for 2P, it is a thing, and I guess you'll hear about it later.
Absolutely. Yeah, definitely a future question. But, 15, 20 years ago, the things you're talking about here, using a voltage indicator with, 3D patterned optogenetics, would have seemed probably as far-fetched as the question asked here about, miniaturized super resolution two-photon. So I'm really glad people were asking.
No, I mean, the whole point, I mean, we spend a lot of our time, every feature, whether it's software or hardware on our microscopes, comes from some sort of collaboration, either really labs that are heavily involved with us, or even just normal everyday users who want to do something different with their experiments. So we really try our best to listen to what people need, and we're not just kind of making features because we can, if that makes sense. Like, we're really trying to tailor what we do for what future needs of experimenters are going to be. So if there is a world out there where we want super res 2P imaging, I'm sure we'll start looking into it. This is just news for me.
That's a great answer. Thank you. I have a couple questions related to optovolts here. So it's great you already have the slide up here. With kilohertz imaging, when you're doing voltage sensor neuronal imaging, what kind of temporal resolution does that give you? Does that give you single spike in all neuron types? Are there any types where maybe that's a struggle? And then do you get waveform information as well?
Yeah. So I will say that it's, cell type does matter. So, some of this actually, I think one of these traces I actually put on here is from a fly neuron. And in a fly neuron, at least when we were imaging it, this specific neuron, you could really only see essentially 1 or 2 time points per spike. So no real waveform extraction can be really done there because the waveform is so tight. What it looks like so far in mammalian systems is a little bit better. Like, you can actually see, the rebound. I am poor in the amount of data that I have right now, so I haven't really reached the point where I've been able to do a lot of averaging across different spikes to see a good waveform. But my guess is that it would be good enough for it in broader spiking neurons right now. So And also I should say that the indicator does matter. So um, the various indicators that are out right now, I didn't make a slide comparing them here, but the indicator kinetics themselves also will low pass filter a spike. So you could potentially intentionally use a slower indicator if you wanted to, if you wanted to see more information. There are also some of the indicators that are better tuned for subthreshold events, for example. So if that's really what you're interested in, and like the indicator you want to use, you really need to compare things like its kinetics and also the range of voltage where it's most sensitive and most linear. We're now at the point, right, where there's enough of them that you can actually pick your faster one. You can pick your slower one if you need to. So yeah, people actually have been doing spike imaging with some of the older voltage indicators at even lower imaging rates, like 500 or so hertz. So it really does depend on the indicator you're using, and yes, also the neuron that you have. So I imagine if it can capture a fly spike, then we're probably good for most mammalian ones.
That's excellent. Thanks for pointing out how important the indicator choice, kinetics, range are in really designing these experiments. I can tell you've worked with a few labs to make those choices. You mentioned a few applications for OptiVolt outside of voltage imaging. I think I remember you saying maybe blood vessel dynamics or blood flow or something like that. Can you tell us a little bit more about what applications this technology is suited for other than neuronal voltage imaging?
Yeah, that's a good question. So I mostly focus on neuroscience related work because that's my training, but there is vasculature apparently shifts quickly enough that you need to image way faster than your typical kind of 30 hertz resonant imaging that people tend to do. I would say though that like you can, even without one of these systems, if you define a field of view sufficiently, you can also you can also nudge a resonance scanning system into several hundred hertz imaging just by defining a smaller strip. The idea behind this is to expand your field of view, keep it square, but you can even push the boundaries of just a regular resonance scanning system. I was also going to say neurotransmitter, still neuroscience related, but those are events that are occurring in the tens of milliseconds kind of range, order of magnitude. So you do need faster than typical resonance scanning to get there. And we do have a little bit of experience imaging with eye-blue sniffer right now, and it does look pretty good. I have not actually seen this work with vasculature imaging yet, but I'm sure there's a lab out there that wants to collaborate with this. But there's even some things like where people image cardiac tissue that needs to be very quickly imaged. So I've never worked with anyone who does that, but they're mostly outside of neuroscience, I think mostly kind of vasculature related, but I'm sure there's someone out there who wants to do something different. Please send me an email.
While we're on the topic of going maybe outside of the in vivo neuroscience imaging that so many of us in this community think about, what about imaging organoids on a two-photon system? Is that a possible application for the technology?
Yeah, we do have a couple labs that do organoid imaging. Organoids are also pretty dense, so like the 2P imaging is pretty good for getting through tissue, even with organoids. I was going to say I don't really know if there's a lot of data out there because there aren't a ton of labs doing it, but there is the Posca lab at Stanford who images organoids on their system, and there's also a group at where are they? Santa Barbara, that has one, and they're also doing optogenetics in their organoid.
Cool.
Maybe I should actually see what their data looks like. I haven't actually. They've only had their microscope for about a year now, so pretty early on. But yeah, it's definitely compatible with organoids if you want to set up. If you put in the effort to have the appropriate chamber under your scope, you can do it.
Awesome. The next question is a sort of combination of hardware and application. So they ask, I'm wondering whether the ETL can be used to extend imaging depth due to working distance limitations for the objective. And then they say, especially in deep brain regions like striatum or hippocampus. So what do you think about this? Can the ETL be used to extend imaging depth to get to striatum, hippocampus?
Yeah, it could, right? So yeah, you essentially define its sort of 0 point manually by telling it, what it thinks 0 is truly by comparing it to, the image you generate, like in just the 𝗨𝗡𝗟𝗢𝗪 path. So no objects in the line there. And that's like the true working distance. And, this is an interesting one because it's not the ETL Z range is not totally uniform from its Z equals 0 point. I don't remember how much it goes above versus below, but it should extend your imaging a few hundred microns. If you are working in a situation where you have like a 3 millimeter working distance objective and you need another millimeter, it's probably not going to be enough for it. But it does actually extend your working distance because it's changing it's essentially changing the collimation of the light going into the objective. So basically, like kind of either converging it or diverging it to adjust the focal point. So yeah, it would work as a focus extender, but I can't imagine you get much more than a few hundred microns out of it. If that's enough, then yes, it'll work. And that also, actually, I should say that depends on the objective as well. So lower magnification objectives will give you a longer range.
Okay, the next question is asking for a better field of view and DOV. I'm not sure if that's a familiar acronym to you, Kevin. Is it referring to the
Depth of field?
Depth of field? Okay, with DOV. So for a better field of view and DOV, if this is your question, feel free to clarify. I'll watch your response. Can we use a customized transmission grading? They mentioned this is cost friendly instead of the more expensive DMD or SLM. How much would using a customized transmission grading compromise the resolution of the image?
Wow. Yeah, I bet you could. I'm probably going to have to follow up to answer this over an email. So whoever is asking this, let me know, and
we'll get back.
I should follow up. Well, I should follow up with our optics people because I'm not actually sure. My guess is that actually resolution would suffer from it, but I don't really have a lot of experience with that. I know that we can use these to generate, for example, multiple points for kind of faster scanning, but I've never actually really experimented with using them for changing depth of field or the like. But yeah, DMDs and SLMs are not cheap. That's you're not wrong in that sense. Let me follow up with that because I'm not actually sure what the end result of that would be.
Excellent. Yeah, we'll make sure we follow up offline there. We have another question here that's really asking, how do you handle brain motion? Of course, there's so many beautiful things about doing in vivo neuroscience imaging, but sometimes, you might see brain motion that makes it harder to interpret your imaging data. Is this something that you deal with online, offline? What are the most common methods that you see, Kevin, for handling brain motion?
Most commonly, it's done post hoc. There are issues with that. So I'll pull this back up here. So a couple different things to consider. So one of them is drift. So if your field of view is drifting over time, yeah, I don't know if that's like stability of the implant or whatever, but that can happen. There's a method you can implement to say move the stage slowly. You can pull images and do a cross correlation on either separate computer or a separate basically set of code that you can write here, and you can tell if the system is drifting in X, Y, and then and feed back to the microscope to move. That's how people generally deal with long scale drift, because if your image drifts out of your field of view, you can't motion correct it. For rapid motion correction, like heartbeats, I mean, even fruit fly brains pulse as well. This is typically done post hoc. Personally, I like to use 3D Volreg if it's a volume. This is part of AFNI. I guess Sweet 2P is really commonly used because it's fairly fast, but I also think that there's a great tool called Advanced Normalization Tools, or ANTs. These are, I highly recommend people look into fMRI, that field, because motion correction and motion and image registration has been they've worked this out really well in the fMRI field, and they've been doing it for longer with more funding, it seems. So a lot of human neuroscience packages work really well with 2D imaging data, because at the end of the day, you're just kind of looking for contrast and these sorts of things. And like I said, ants can do nonlinear registration and motion correction as well. If you have a lot of motion in Z, typically the best method for dealing with that is to, if you can, image a volume. So if you cover the whole area, post hoc volume registration is pretty good as well too. You do sacrifice imaging speed the more slices you collect, but I used to do whole brain imaging in Drosophila, and that's taking, that's imaging across a couple hundred microns in both X, Y, and Z. And yeah, it's challenging to do that quickly, but if you capture everything, you can always fix the motion later.
Yeah, these are great tips. A lot of ways to handle motion, and it sounds like some of it boils down to acquire more. Make sure you have the data to be able to do the corrections later. To the audience, we do have time. We have another 8 minutes in this session, so feel free to put your questions in, or if there's a topic that was introduced in the presentation that you just want to touch back on, happy to take those requests. But I think our next question is maybe inspired by what we see on the slide here, this closed loop, all optical interrogation of neural circuits in vivo. I think that's the title of the paper. So the question is, can holographic optogenetic simulation be used in a closed loop manner? And if so, based on what input? Is it based on the neural activity, sort of the live imaging as the input? Is it based on some TTL inputs or other aspects of the experimental setup?
Yeah, good question. So for what's typically done, so like the easy way to do this sort of thing is synchronization with like a behavioral setup. So if you have, and that would be just TTL pulse synchronization. So the system that defines your hologram can essentially wait for, and let's say, if your mouse licks like the sugar port or whatever, and your behavioral apparatus is set to trigger optostim anytime that the mouse licks the thing, or if it's a fruit fly, if it turns a certain amount, I know someone who's doing this for navigation work. If the fly turns a certain amount, that triggers optogenetic stimulation. That's the most commonly most commonly implemented way to do this, and that's just with electrical integration. If you do want to do something like real-time analysis of, say, a neuron, that's where PrairieLink would come into play. We don't have an online measurement tool yet, but essentially what you can imagine your PrairieLink setup, getting an image and setting a threshold for a certain, certain just amount of signal, right? If you define an ROI and wait until it crosses the threshold, and then you can have that send the command to run stimulation through PrairieLink. That's a possibility. I don't think I know anyone who does do that, but it is certainly something that I think some people would want to do, right? So once a certain pattern of activity occurs in the brain, then it will trigger it. That's going to be more custom code writing, but it shouldn't be too difficult. PrairieLink works in a bunch of different languages. I myself am partial to Python, but I think you can even run it from like a shell script if you really want to. It's like pretty simple to get working.
So you mentioned being able to define an ROI, right, and set some threshold. Can you define a complex ROI with the PrairieLink system you're describing? So could you have individual somas, neurons, regions of interest set as your threshold? Or could you set threshold of, here's 10 neurons, I want 5 of them to reach my threshold, and there's my trigger. So what kind of complexity can you introduce here?
I mean, if you can code it in, MATLAB or Python, you can do it. So once you've PrairieLink basically just exists to pull data from the scope or send commands to it. Once you're doing that, you can write whatever, you can use whatever package there is out there that you want to use for ROI definition and threshold setting. It's been a minute since I've used some of these, but there are a bunch of great Python packages out there. So if you're willing to put in the time and effort to write this, you can do whatever you need to. I mean, the limit is your imagination, I suppose.
That's a pretty inspiring note, I think, to conclude the session on here. Thank you so much, Kevin.